Thank you, everyone, for joining the first seminar of the 2026 IQB Spring Seminar Series. You can see the lineup here for this term. Well, we have an interesting set of seminars lined up, and our first speaker for the series is Kate White from Allen Institute. Welcome, Kate. Kate completed her bachelor's degree in biology from the University of Illinois, Urbana-Champaign, and her PhD in pharmacology from the University of North Carolina, Chapel Hill, where she worked on identifying biased kappa opioid ligands for probing signal transduction pathways. She then shifted gears into structural biology and joined the Ray Stevens lab at the Scripps Research Institute in San Diego as a postdoctoral research associate. And then when the Stevens lab moved to University of Southern California at Los Angeles, she moved with the lab and joined the Bridge Institute. She was first a postdoctoral researcher and then an assistant professor of research. And in 2021, she became an assistant professor at the Department of Chemistry at USC. During her time at USC, she also led the Pancreatic Beta Cell Consortium as its director. The consortium is a community of scientists, clinicians, and engineers working together to develop new methods for understanding beta cell biology and treating diabetes through cross-disciplinary approaches, as well as creating a spatiotemporal whole cell model of the human pancreatic beta cell. In fact, I was introduced to Kate through the consortium where I was involved in supporting the data infrastructure for the consortium's whole cell modeling initiative. We were actually introduced by Helen Berman. I think she has joined the seminar today, the PDB's director emerita, who has been a mentor to both of us. Kate also used to organize the annual meetings of the Beta Cell Consortium at USC, where on one hand we had very focused and intense scientific sessions, and on the other hand, we had a lot of fun team building and social activities. Thoroughly enjoyed these meetings, Kate. In 2025, she moved from USC and joined the Allen Institute for Cell Science in Seattle as Associate Director of Quantitative Cellular Imaging. This is a new and exciting chapter in her career, and I wish her all the best in her new endeavor. Today, Kate will be talking about her work on quantitative structural cell biology to map cellular functions. Kate, I wish this seminar was in person. It would have been very nice to catch up, but the weather unfortunately made it difficult to travel to New Jersey. Nevertheless, I'm glad we are able to have this seminar online. You're able to give this talk online for the Rutgers community. Thank you very much for joining us today. Over to you, Kate.
Thanks a lot for the introduction and for the opportunity to share the work that my group has been working on for the last few years, or several years, I guess. Let's see if I can get this started. There we go. So yeah, thanks again for the introduction. Mostly what I want to talk about today is a story about how we are using quantitative structural cell biology to really understand cell function. So actually all the work that I'm going to talk about today was done at my lab at USC, and most of my students are still active on site at USC. If I have time at the end, I'll give you a little hint of what I'll be working on at the Allen Institute. So one of the main kind of model system that we've been focused on is pancreatic beta cell biology. So I want to just give a little bit of biology background before I dive into some of the imaging strategies that we've been developing. So pancreatic beta cells are shown in the green. So these cells secrete insulin. They're the only cells in our body that secrete insulin. They live in pancreatic islets. So this is a cluster of about 1,000 cells that all kind of work together to secrete hormones that really help regulate a lot of different aspects of our biology, but we're going to focus mostly on blood glucose homeostasis. And what happens when there's beta cell dysfunction is basically these beta cells are unresponsive to glucose, so they aren't secreting insulin, and insulin is not maturing within the cell as it should. So that is a big sign. Insulin, if insulin doesn't mature, basically it's almost like you're secreting nothing. So this is a part of the biology that we're really interested in understanding better. And ultimately, our treatments for diabetes, the still main kind of treatments are insulin injections. There's been a lot of work on some stem cell derived cell therapy, but it's still pretty experimental. And ultimately, what's missing from this is that we don't really understand enough about the fundamental beta cell and islet biology. So that's what my group has mostly been focused on, in really terms of understanding how does the cell regulate insulin maturation as well as secretion. The other thing that we've really focused on is thinking about single cell analysis and really trying to understand cell heterogeneity. This ends up being pretty important for islet biology. And so, just for example, the bottom left here shows kind of just a classical biochemical assay. Maybe here we can think about insulin secretion. We'll have some kind of, we'll be looking at an averaged effect over hundreds of cells, and just take the average and think, okay, that's, roughly how much insulin is being secreted. But sometimes, there's some really interesting biology that happens where really not all the cells are acting the same. So in this video, we're looking at calcium responses in an islet. So all the blue cells are beta cells, and the green is flashes of calcium. So we can see that this isn't happening uniformly. Certain cells kind of start this trigger. Certain cells have a higher intensity of calcium release, and this is really linked to insulin secretion. So we're wanting to understand these differences and how these cells are functioning collectively. So basically, the what we're really wanting to capture is the reality of these cell phenotypes. So sometimes what ends up being really important for the biology is what's kind of maybe not what's happening at the average. We can have different kinds of responses, and so we're really interested in capturing this single cell heterogeneity in terms of really thinking about how the collective action of an islet is working. So to do that, we need to do a lot of single cell imaging. Now, the thing that we're really interested in capturing in this imaging is how insulin is stored, matured, and secreted from the cells. So I just want to cover a couple of basic kind of key principles that we know we want to look for. So on the left is kind of a map of where insulin's coming from. So it's budding off from the Golgi here. We have these immature secretory vesicles that once they kind of bud off from the Golgi, they actually undergo an acidification process that allows for proinsulin to be cleaved into insulin. And once this happens, insulin is able to, there's so much insulin inside of these secretory vesicles that it actually crystallizes. It forms a protein crystal inside the secretory granule. And this is those are two of the main hallmarks for maturation: acidification and dense core formation. And again, if that acidification doesn't happen and proinsulin doesn't get cleaved, then proinsulin cannot engage the insulin receptor. So this is the process that we're really wanting to dive into, and in terms of really thinking how the cell is regulating this process, because we know it's defective in disease, but we're not really sure why. The other thing that I want to point out is that not all insulin granules are really the same. So over the last 5, 10 years, there's been a ton of data that's come out to suggest that there's differences in their protein composition, their lipid composition. The behavior of old and young insulin granules is different. different subpopulations of granules respond to different stimuli. And then again, some of these subtypes of the granules are associated with disease. So we're just really interested in trying to learn how do these vesicles end up with different lipids, different proteins, and what's regulating that process in the cell. So, luckily for us, this has been a heavily studied area, especially in the pharmacology world. So, there's an abundance of chemical probes that we can use to study insulin maturation. So, the world knows of a lot of different ways that we can force a cell to secrete insulin. Ultimately, the kind of key signaling pathway that's similar is an increase in calcium, which allows for the secretory granule to engage the snare machinery and secrete insulin. And so there's a lot of different ways that we can ask a cell to do that. And so ultimately, sulfonylureas you may have heard of, but ultimately this is really just leading to depolarization of the membrane and allowing for calcium to spike. Glucose itself can act through the metabolism pathway, and then we can have a glucokinase activator to overdrive metabolism. This activates a bunch of different signaling pathways, but also calcium. And then we can also activate incretin receptors. So this peptide here is actually GLP-1. So one of the ways that it works is by engaging cyclic AMP signaling, also leading to calcium flux. And then the GPR40 is another GPCR that activates the PLC signaling pathway. And all of these lead to insulin secretion. But they, as you can see, they activate different signaling pathways within the cell. So we're really curious how these different mechanisms of insulin secretion are actually impacting insulin maturation within a single cell. So this actually had not been well understood to this point. So we are going to be looking at this through trying to understand the density of these secretory granules. Again, if we have this dense core, we know that maturation is happening. And we're also going to be monitoring pH. So we'll use density and pH sort of as our readout of maturation of insulin. We really want to dive into the timing and dynamics of this process, and then really try to understand how these different chemical probes impact the types of subpopulations of secretory granules that are there. So again, back to the single cell aspect. When we first started this project, and this was through the consortium that Brenda had mentioned, I was really trying to find the best way to get at these questions in a hopefully a high throughput or the easiest way possible. And ultimately, what I was really ended up being focused on is non-biased imaging. So this, some kind of method that has non-biased imaging, no tags required, meaning I don't have to know necessarily all of the cellular organelles that I'm interested in observing. I knew for sure we wanted to look at insulin granules, but I didn't know what else was going to be important. So I wanted a method that would allow us to image as much of the cell as possible without having to tag certain proteins or organelles. I wanted it to be relatively high throughput in that I could screen a lot of different drug conditions, look at different time points. And then the most critical aspect really was to be able to look at the entire cell. So we could actually do a quantitative analysis of the cellular organization, polarity, how many insulin granules are there, where are they, how dense are they, to really get at quantitative numbers to see how these different conditions are really changing the organization of the cell. So we landed on soft X-ray tomography and collaborated heavily with Carolyn Larabel's group there at the Lawrence Berkeley National Lab. So this ended up really being a perfect microscope and technique for basically the goal of what we wanted to achieve. So this is a very special microscope. A lot of people haven't heard of it just because there's not a lot of them. This is really the only one of its kind in the States. It's a full field transmission X-ray microscope. Light source for this particular one is synchrotron radiation. And the optics we can get to was about 35 nanometer resolution, which is controlled by these two zone plates here. And we can from the time of actually starting imaging to getting a cell reconstruction, a full 3D reconstruction, is honestly a little less than 15 minutes. So that's pretty fast when you think about serial section EM kind of techniques. And then again, this is unbiased. We can image these secretory granules in the context of the whole cell. So just a little bit more about the contrast mechanism because it ends up being pretty important for interpreting the data. The key for why this works so well is that we're basically imaging at the water window. So what that means is biomolecules containing carbon and nitrogen are going to be absorbing more X-rays and water is absorbing very little. So essentially, water is invisible, which is why we're able to image through the entire cell and not have to do any sectioning. The other aspect that's really important here is that absorption of the X-rays follows a Beer-Lambert law. So basically, the more concentrated in biomolecules a specific voxel is, the more absorption that we see. And that's linear. So what's shown in this graph is basically this is glass capillaries full with known amounts of BSA. And when you basically plot the known concentration of BSA to the absorption, it is in fact linear. So we can use this as a strategy to actually identify a range of secretory granules based on their density, a range of maturation. So this is really kind of the perfect kind of method for really wanting to look at these small particles within a single cell. So what we have here, this is a pancreatic beta cell. Um, and all of these small puncta are secretory granules. And so you can see kind of this range here of different sizes and different densities of secretory granules. So I'm going to just show a video right now that just dives into what this data looks like a little more deeply. So the cells are frozen in a glass capillary. Right now we're just watching this is not a time lapse. This is an orthoslice by orthoslice view of the single cell in a glass capillary. And we get, isometric imaging. And now we can see we've segmented this cell, so you can see the nucleus, mitochondria, and insulin secretory granules. And what's great about this is that, when we get this mask for where the organelles are, we can do a lot of analysis in terms of really understanding where the secretory granules are. Are there clusters of secretory granules? How close are they to other parts of the cell? But most importantly for us, if we bring in that linear absorption coefficient information, which basically means the insulin granule density, we can actually color the secretory granules by their densities. So the yellow in this case are the highly dense granules, and the blue are less dense. And you can actually see in this video that there are pockets or clusters of more dense granules. So we're still kind of investigating if this is, hotspots or areas where active maturation is ongoing. We're trying to understand if there are cellular neighborhoods that are really regulating this process. So my student Anish Deshmukh, who will be defending in 2 weeks, did a pretty big screen with using soft X-ray tomography to really get at that question that I outlined in the beginning for which of these signaling pathways are responsible for insulin maturation. Again, we know they're all responsible for insulin secretion, but do they equally impact the maturation of insulin? Because ideally, we're going to treat diabetes, we're going to want to be able to enrich for insulin granules that have a lot of mature insulin within them. So this is just some of the biochemistry that he worked out. What's shown in this graph is just the amount of insulin secreted from the cells during this treatment that he does. So we can see that basically treating with the GLP-1R agonist or a GIP agonist, which is very similar to GLP-1R, basically activating the cyclic AMP pathway, leads to the most insulin secretion. And that was not surprising. This was expected. But when we looked at the secreted insulin to proinsulin ratio, this actually gives us a sense for which of these conditions might actually be enriching for mature insulin to some extent, and that we can look for insulin to proinsulin. If there's a high insulin to proinsulin ratio, we know that some maturation is occurring. And again, it looks like probably the incretin signaling pathways seem to have the most robust impact on this. But there also could be some just from activating glucose more directly. And then I want to point out that the gold is sulfonylurea. So this is just basically activating the or depolarizing the membrane of the cell. And we see maybe a little bit of an effect, but it's kind of hard to tell in this assay. It's probably not sensitive enough. So this is where we move on to really looking at what's happening inside the cell using soft X-ray tomography. So ultimately, what Anish found in this case is that all of the drugs really had a pretty unique impact on the secretory granule maturation. So what's shown in the top is just representative examples of kind of how dense the secretory granules are and their relative size. So we can see that lipid signaling here in purple, that's the GPR40 agonist, really led to the largest and most dense granules. And then if we look at extendin 4, which is similar to GLP-1R or GLP-1, and GIP, both incretin peptide molecules that activate somewhat similar pathways, they actually have pretty different responses. So GIP, we see that there's a lot more vesicles in the cell, but they're not as dense as what we see with um, lipid signaling and GLP-1R activation. And this is just a quantification of that data. So really, whenever we're kind of over activating metabolism, that's what's shown in green, a glucokinase activator. Even lipid signaling and then incretin signaling seem to have the biggest impact on the density of the secretory granules. Whereas if we just if we depolarize the membrane, we don't really see much of a change compared to glucose. So at this point, it really seems like depolarization alone does not impact secretory granule maturity. And then those treatment conditions that do seem to impact the density of these granules, it's clear just from looking at data that they do seem to be having some unique impacts on the maturation. So Anish wanted to dive into this a little bit deeper. So this is just another way of really looking at this type of data. So what we have in these graphs, the lac is the you can think of as basically the density. So we look at this graph over on the left, the further on the right we are, the more dense those granules are. And then the diameter is shown on the other side. So we can see basically that if we just depolarize the membrane, we have a kind of a minimal effect on the density and on the diameter. If we overdrive metabolism, we do see this shift in density, which is pretty obvious in this graph, and the diameter actually slightly decreases, which makes sense because the insulin is condensing and forming that protein crystal. Now, when we treat the cells with the lipid GPR40 agonist, we see an increase in the density of these granules, but we also see an increase in the diameter of these granules. So, which suggests that that protein crystal, is actually larger in these cells. And then for the two incretin cases here, this graph nicely shows the difference in the density. So there's a bigger effect on the density with extendin 4. And in both cases, the granules are pretty small, so they're tightly compact. The next thing that Anish really wanted to investigate, so he was doing these experiments on a beta cell line, which is great for doing biochemistry, not the best for really wanting to study disease, though. So we've done a bunch of experiments now to validate this in primary beta cells, and he's done some with human beta cells as well. So what we have shown here is a mouse beta cell. And even before he quantified the data, you can tell that the density of the granules treated with extendin 4 there's a clear difference. So we get very similar data in our primary cells. And then I didn't show all of this data for all the other conditions, but basically we can look, this graph is showing us basically if we think about the position of the granules within the cell, the most dense where the most dense granules they seem to follow kind of an equal path throughout the cell, no matter where they are. This is distance from the membrane. So that, there's not really like a specific area in the cell that they're at, kind of all the way throughout. We see this increase in density evenly throughout the cell. So what Anish has been working on more recently has been really trying to understand more about what is actually changing in these secretory granules. And what's great about the soft X-ray tomography work is that we can do the quantification of what's happening in a single cell and really have an understanding for how the whole cell is reacting to this. But the resolution on what's happening in individual secretory granules it isn't as good. So he has shifted to cryo-electron tomography to really get a better sense for what's actually happening inside of these secretory granules. So this is just looking at similar kinds of conditions, but with much higher resolution. So what we've got here on the left is our basal condition. And you can actually see in some of these the crystal lattice of the insulin dense core. So we can see that in basal conditions, but ultimately a lot of times without treatment, we don't really see that really dark crystal or noticeable crystal lattice. We'll see some kind of fuzzy, dense core. And then when we treat with extendin 4 here, again, this is our GLP-1R agonist, you can see that there's a nice crystalline aspect to this dense core. Very obvious here with the lipid signaling molecule. You really clearly can see that this is a protein crystal. What's interesting at this point is that we also start to see some fibrils along the edge of the granule. And so at this point, we think that those are probably pro-insulin fibrils, but we're not entirely sure what that is yet. Then there was another control that we did that I want to talk about because it was really baffling for quite a while. So we had treated the cells also with bifilomycin to prevent granule acidification. And when we did the soft X-ray tomography data, we saw that they were still pretty dense, which was really puzzling to us. So when we looked at this with cryo-electron tomography, it was really interesting because we saw this huge protein aggregate. That's what we think it is. We don't entirely know for sure what this is, but basically, this is most likely insulin. And then because the pH is dysregulated in these granules, we start to see just these huge aggregates that was leading to that dense signal that we were getting in soft X-ray tomography. But ultimately, the cells are quite sick in this case, but I just wanted to use that as an example for why using multiple imaging modalities really is important for trying to understand what's actually happening inside these granules. And then this data to the right here is a rodent model of type 2 diabetes. And so we've done a bunch of additional studies with human cells in health and disease. But what I wanted to point out really is that even in a disease case, this insulin, it does, there is an insulin crystal in this granule, but you can start to see in the center now that there is a lot of fibrils. And in this case, this is likely amyloid. Uh, so that's one of the kind of problems that can occur in diabetes is this formation of these amyloid aggregates inside the secretory granule, which leads to a lot of dysfunction in terms of the maturation and secretion of insulin. So this at this point, another student in the group really kind of looked at all of this data and wanted to think more deeply about how we can classify these different subpopulations of secretory granules with the data that we have, because we know that insulin maturation is dysfunctional and disease. And we know certain things to look for in terms of what a not healthy secretory granule can look like. And we know that some drugs work better than others, but why? We're really still trying to get at why is that. And so we think that it's likely because there's a shift in once you treat the cells with drug, there's a shift of what kind of subpopulations exist, and so we really wanted to dive into that more deeply. So Kevin and my group really kind of took a very creative approach to get at this. Let's see, I already kind of mentioned some of this, but just to remind you again, there's this kind of shift in the protein and lipid composition, as well as kind of the size and density of these granules. So the next two stories I'm going to talk about are related to this aspect. So what Kevin did was really look very closely at Anisha's soft X-ray tomography data set and think about how he could pull as much feature information out of each one of these secretory granules. So before we kind of just looked at what's the mean density. We didn't really think too much about shape or position in the cell as much. So that's what Kevin was really going after here. So this histogram is basically a distribution of the density information for this particular secretory granule here. So not only do we want to look at kind of the mean density, we also wanted to get a sense for basically the shape of this curve. So by looking at the kurtosis and skew, that gives us an idea for the shape of this curve, because you can imagine that perhaps one secretory granule or two secretory granules could have the same mean density, but one could maybe have a very dense small core, and another one could have a slightly more diffuse, larger core. But really, those are two kind of different, those are two granules that are in different states of a process. We really wanted to be able to try and capture that information. So including kind of the standard deviation, and then the shape, diameter, and then how far away are these granules from the plasma membrane or other organelles within the cell, to really try and featurize the complete story of what's happening with these secretory granules. So basically, Kevin took all of those features and all of Anisha's data, and he ended up using these features for k-means clustering. And he set up a UMAP basically to try and sort out how many clusters of secretory granules that there are. And so he basically found five stable clusters that can kind of correlate to less mature granules and more mature granules based on the density of the granule as well as position within the cell. So when we then look at how these different stimulations are kind of impacting these, relative the populations, it's quite clear that there's a dramatic impact. So in the case of no stimulation, we can see that this population one, which was the least mature, seems to be predominant. But what we see as soon as we start adding glucose, and then GLM is the sulfonylurea, so that's the membrane depolarizer. Ultimately, these two profiles look very, very similar. But when we start to overdrive metabolism, that's what GKA is doing, we can see a pretty clear shift in the subpopulations. And then lipid signaling really enriches for subpopulation 4. And then GIP and extendin again is like a GLP-1R. We can see these profiles. So I'm just going to play this video, which gives you a sense for basically he's colorized all of the granules to match the subpopulation of granule that that they were classified as. Um, and so, you can, especially with the colorization of those granules, you can really start to appreciate the different features and the different kind of impacts that these different signaling molecules have on kind of the identity of these secretory granules. Okay. Yeah, and so kind of what's next here is diving deeper into what's different about these granules. So there's a lot of plans for doing follow-up proteomics and lipidomics to really understand more about what's physically changing in terms of protein and lipid identity between these granules. So I want to just kind of summarize what's we've done with the density mapping strategy. So it's clear that these different signals are having specific effects on the secretory granules. We see these density changes can occur within 1 minute of stimulation. So I didn't include that data. The data I showed was 30 minutes after stimulation, but we can see that this we have a lot of data sets that look as early as 1 minute. So that's a pretty quick amount of time to have a density change. So to us, that really indicated that pH, pH would be a fast change. This a pH regulated aspect? And then we also see size changes within 30 minutes. So that is a longer time period. And if a secretory granule is growing, if it's getting bigger, perhaps that means there would be some lipid exchange to allow that to get larger. So I'll talk about that part at the end. The other thing I want to mention is that we have extended a lot of the structural analysis to the mitochondria organization. And it's very clear that the mitochondria are responding to these drugs and really helping to facilitate the action of maturation as well. Okay, so then next thing I want to dive into is really this relationship between the density and the pH. So this is again how we were measuring the density. And now we're going to do similar experiments, but focus on really monitoring the pH. And so a very clever student in my group did these experiments. Basically, he redesigned a pH probe that was going to be sensitive enough for monitoring the pH of individual secretory granules within the cell, which ended up being a little tricky, but this is basically we can express these in the secretory granule and then do all of the imaging. So the way this pH probe works is it's a fret sensor. That in the case, it's a flim fret sensor. So we're actually monitoring the lifetime signal of this sensor. And essentially, when we have a shortened donor lifetime, that we're getting a fret signal. But when proton is bound, we'll have no fret signal. This changes the lifetime of the molecule, which we can monitor. So we can get a sense for if there's more protons, this is a signal we should get. And then we can actually use this to calculate what the pH is. So in this case, if we're analyzing this data set, this again would be kind of donor only, 100% protonation, and then max fret 0% protonation. We can use this percent protonation to really give us a readout of the pH. So what Wen did was express these in cells and permeabilize the cell membrane and add known amounts or known pH into the buffer so we could make this nice pH calibration curve. So this is the curve that he used to basically quantify the pH in each one of these individual puncta. And so this is what some of the data looks like. This is just a short video so you can actually see some of the dynamics of these granules moving around in the cell. So we did 3D live cell pH determination at basically for each secretory granule in the cell. And the first thing that he noticed was that when we look for the most diversity or the most kind of heterogeneous area in the cell was the membrane, the granules near the membrane. And this kind of makes sense because You have really mature granules going there, but then they're also secreting at this point. So we have a pretty broad range of what we can see. So the peripheral granules have a diverse pH, and then the perinuclear ISGs are most acidic. So now then Wen did a couple of treatments to dive into this. So again, extended 4, he's looking at this is our GLP-1R activator. When he adds this drug, and in this case, he's not adding glucose, he's only adding extended 4. So this will not lead to insulin secretion because you need to co-administer this with glucose to get secretion. So all he's doing is activating this pathway and looking at what's happening to the pH of these granules. So they're not being secreted. And what we see is that there's actually a pretty big shift in more acidic secretory granules after stimulation. So this graph at the bottom here is really showing you the difference after stimulation compared to what's shown at the top is just showing you with baseline, and then after 15 minutes, and then after about 40 minutes. And so it's very clear that there's a little bit of a reduction of this medium range, and then a big increase in the acidic granules. So this is in fact acidifying the granules, which just confirms what we suspected from our other data. Wen did another experiment where he depolarized the membrane with potassium chloride. This is going to work pretty similarly to the sulfonylurea experiment. And in this case, we're getting a big secretion or release of those acidic granules. And then there is some movement, there is kind of an increase in these less acidic granules, likely just because of the movement of the granules within the cell. But what I want you to look at here is kind of this range of granules that are being secreted in this case. Because on the next slide, I'm going to show what happens when we stimulate the cells with glucose instead of depolarizing the membrane. And in this case, what we see is that there is a more narrow range of secretory granules that are being secreted. So it's only the most acidic granules were secreted, and we actually see some acidification, an increase in some of this mid-range secretory granules, which definitely tells us that these different mechanisms are mobilizing distinct subpopulations of these granules for secretion. So yeah, basically this data is in line with the time courses that we saw with soft X-ray tomography. So it's clear that there's certainly a link between the acidification. The limitation for this currently is that we've only labeled the secretory granules. So what Wen's been working on is really to try and label other structures in the cell to really understand where this pH change is happening, how quickly is this, and how do other organelles perhaps contribute to this process. The reason I keep talking about other organelles is because the field of cell biology has really exploded in understanding that communication between organelles ends up being really important for normal cell functions. So there's a lot of this has been documented that there's transfer of biologically relevant signals, intracellular lipid trafficking, a lot of lipid metabolism, a lot of calcium signaling that happens between two different organelles actually physically communicating. And one of the things that we noticed pretty early on with some of our studies was that we were able to observe that in the conditions that enrich for the maturation of insulin, we saw noticeably more secretory granules in contact with mitochondria. And what I'm showing up here is actually some quite pretty old data. This is from Peter Butler's lab, actually, the one on the right here, where so folks have in fact seen that secretory granules do come in contact with the mitochondria, but before, no one really knew what to make of this. But because we were able to look at the entire cell, we could actually quantify how often this happens and find out that in fact, This is statistically more significant of an event in conditions that are enriching for maturation. So what's shown in this graph down here is this is the mitochondria in one of our pancreatic beta cells, and we can actually it's colorized also by its density, and that we can see wherever a secretory granule is in contact with the mitochondria, the density, the molecular density of the mitochondria is different, which indicates that there may be some, yeah, so there's actual physical contact, but then there may be some signaling molecules there. What we found out from analyzing all of our soft X-ray tomography is that the larger vesicles are in contact with the mitochondria. Those contacts occur more frequently near the plasma membrane. These contacts are enriched within 5 minutes of stimulation. We're starting to wonder if there's some kind of lipid transfer occurring between these two organelles. My students have, really been trying to chase this down and trying to understand when in the process of an ISG life cycle does this happen. The other thing that we've noticed with a lot of the cryo-ET studies is that we've noticed these protein densities. And what I'm about to show is sort of, this is possible hallucination. But it's clear there is definitely some kind of protein density here. But when we looked at this, and we've seen it many, many times, and when we see this kind of shape that my student sort of outlined here, it does look a lot like a lipid transfer protein. So in this case, this was from another paper. Pietro Di Camilli's group published this, and this is just kind of what a lipid transport protein looks like. So I'm not saying that's for sure what this is, but it does make us really curious about what is actually going on here. If there's material being exchanged between the mitochondria and secretory granules, and if this is enriched in right now, this is just kind of correlation that we see this happening more when the mitochondria or when the secretory granules are maturing. So we're really curious to dive into this and really figure out mechanistically what's happening. But at this point, we have more questions than answers in terms of what's actually happening on that part. And then my student, Rachel, has been really trying to get a sense for when in the life cycle are these secretory granules in contact with the mitochondria. So she is using this approach to do a pulse chase experiment. So we have insulin with a snap, basically a snap tag on it. So basically we can label the secretory granules at certain time points and see kind of where they are in the cell. So here, these are 3-hour-old secretory granules. We can see that they're, haven't really moved to the exterior part of the cell yet. And then 8-hour-old and 12-hour-old. And when she really quantifies this data, what she's found is that basically the younger secretory granules seem to be in contact more with mitochondria. So really to kind of wrap this up, we're still kind of investigating this lipid transport and really trying to understand the pH of these secretory granules as they're moving within the cell, and see if other similar kind of contacts occur in similar other secretion cells. The llast thing, I hope I have time for this. I want to show a video of, so Brenda alluded to this, that we've been doing really trying to think about multi-scale spatial temporal cell modeling. So this is just some collaborative projects that the group has been really invested in. And one was really thinking about Bayesian meta modeling, but the video I want to show you is something that I've worked on with Helen Berman quite a bit on an interactive VR experience of the beta cell signaling. So before we watch the video, I just want you to understand the visual language that was developed. We're using tetrahedra to represent these proteins. So here's GLP-1R with all of the different um, representations. So we're representing it as tetrahedra because this translates well in a VR system, and it does a really nice job of showing symmetry of the molecules. So I hope you can hear the audio. I might not play the whole video or skip parts.
Insulin is
Running
key to
behind.
managing metabolism and blood sugar levels. Inside your body, special cells create insulin, then store it in cellular pockets known as vesicles. As the vesicle's environment fills up, the insulin molecules will naturally bump into each other and interact. First, individual insulin monomers pair up, forming dimers. Then, dimer pairs will bind as well, with 3 of them coming together to form a hexamer. Gradually, many hexamer groups will tile and pack together into an elegant lattice structure, an insulin crystal. As this crystal forms, the surrounding vesicle travels through the cell, docking just underneath the cell membrane. When the cell receives the right hormone signal from the bloodstream, a cascade of protein messaging is activated. Several proteins bind and exchange molecules, passing the message along the cell's surface. This message eventually reaches the vesicle, prompting them to start fusing with the cell membrane and exposing their insulin cargo. The exposure to the outside dissolves the crystal back into individual insulin molecules and releases them into your bloodstream. The released insulin will travel and spread through your body, signaling other cells to absorb glucose, lowering your blood sugar. This is just one of the many small miracles happening all the time within your inner space.
Okay, and so yeah, that was a project that I'd been working on with Helen Berman and Alex McDowell and Carl Kesselman at USC. All of the other work that I was able to show today was from my group, shown in the picture, particular Anish, Kevin, and Wen, who had just graduated. And at this point, I'd be happy to take any questions from the audience.
Thank you very much, Kate. That was a beautiful talk about the insulin secretion, the granule maturation. I would let's open it up for questions. There's one question in the chat box. In Smriti Dikshit asks, in soft X-ray tomography, how do you tell the ISGs apart from other granules that may be present?
Yeah, that's a great question. So we did a lot of experiments to make sure that we were only capturing dense core secretory granules. So rather than, there's a bunch of trafficking vesicles. So we looked at cell lines that had insulin knocked out, and we didn't see any puncta or any features that look anything like what we were labeling as insulin secretory granules. We've also looked at a lot of different cell lines, and only cells that have a lot of dense core secretory granules are going to show things like that. So we can also, we looked at alpha cells in the islets, which have glucagon dense cores, and we can, did a lot of work to make sure that we can tell the difference between those. But in insulin or in beta cells, the only dense core secretory granule would be the insulin.
Okay. And Dennis asks, thank you for the presentation. I noticed that at one point you used a rodent model of T2D, and I'm wondering why that was used instead of a T1D beta cell model. My understanding was that T1D is associated with dysfunctional beta cells, whereas T2D is associated with dysfunctional or impeded insulin receptors.
Yeah, that's a great question. In both cases, for type 1 and type 2 diabetes, insulin maturation is defective in both cases. The reason that we chose the type 2 diabetes model, honestly, was just because it was easily accessible to us. And what it is actually challenging to study type 1 diabetes secretory or beta cells because that their health is so poor, it was difficult at the time for us to get enough beta cells to actually image. So that's a good point. But in both disease cases, there is dysfunctional insulin maturation, not just actual, but there's a second part of type 2 diabetes that is you're insulin insensitive, but the cells are also still dysfunctional. So we were just trying to understand what's actually happening inside the secretory granules in that case.
Thanks, Kate. I had a question. So when you study the density, size, and pH of these granules, can you provide some insights into the what happens downstream? Does it affect secretion? Does it affect anything else in terms of whether it is larger, whether it is denser, or depending upon the pH? Of course, I guess interaction with other organelles matters, but other than
Yeah,
that.
and so I definitely think the interaction with other organelles is really important in terms of getting the right kind of signaling molecules on the granules so they respond at the right time, can engage with the snare machinery. But I guess in certain cases where we know the pH is off, or when we know that there's a lot of this kind of amyloid protein around, what we can actually see. Is that the actual structure of the secretory granules kind of starts to implode. And so we're actually having kind of destruction of the secretory granules before they even have a chance to secrete. So that creates a lot of cell stress as well.
Okay, thank you. Stephen, you have a question?
Yes, great talk, Kate. Thank you very much. I appreciate it, and thanks for showing the video at the end. But I think just coming back to Dennis's question about type 1 versus type 2 diabetes, the endpoint for type 1 is no beta cells whatsoever. They become completely destroyed as the patients become completely insulin dependent. So understanding what's going on in type 2 is actually more relevant to the biology. We need to, of course, understand what signals are governing the destruction of beta cells in type 1. But that's the minority of patients. It's a terrible disease, and I don't want to downplay that, but it is not where there's the medical need is substantially unmet. Insulin, particularly insulin pumps, are very with sensors are very effective, and they're also designer insulins that make it easier for patients to manage their blood sugar. So I do have a suggestion for your video, which I liked very much, and I like the representation using the tetrahedra. That's to as you talk about the hexamers being extruded into the circulation, the rate at which the hexamers dissolve determines how rapidly the insulin has its effect on blood sugar. And some of the designer insulins that are now being sold by Novo Nordisk, by Eli Lilly and Company, have long-lived hexamers, meaning gradual release of the insulin biological activity, and others have insulins that have been engineered for the hexamer to fall apart almost immediately. So you have very rapid onset, and you could just by adding a few more frames to your movie, you could actually depict both of those aspects of management of diabetes, whether it's type 1 or insulin dependent type 2 diabetes. Things to think about. There's an infinite number of things you could do, but certainly things to think about. Thank you.
I love that suggestion because the structure of insulin, those molecules were developed just from looking at the structure of insulin and thinking about the hexamer breaking apart. So yeah, that we had talked about that, but hadn't gotten that done yet. But yeah, in terms of patient education, I think being able to watch this video and understand why you're taking this medicine sometimes and why you're taking that medicine, it really would be helpful, I think. Yeah, that's a great point.
And in the global, I think some of the articles in the global health pages in PDB 101 actually
Yes.
explain this in detail, so you
Yes, I
can
have
borrow
actually
from
read
there
it.
freely, of course.
Yeah, yeah, I have. Yes, it's really well described there.
Yeah.
Yeah. Yeah, and it's a
Thank
good
you.
idea to use these videos for patient education. I guess that's useful.
Yeah. Though there is a regulatory process that has to be uh,
Approved.
honored to actually move things into presenting to patients.
Mm.
Something to be aware of.
Yeah, thank
Well,
you.
we're beyond time. Brenda, thank you very much for doing the honors. Kate, thank you very much for giving a terrific virtual seminar. You had, at my last count, you had 53 people online.
Great. Thanks for the invitation.
So congratulations. It was a great talk, and
Yeah, great talk.
I'll look forward to hosting you in person at Rutgers one day. I made a mental note not to start the January 2027 seminars until February after this little experience. So
Yeah, the last couple of
congratulations.
years we escaped the winter storm of this nature,
Right.
but
So congratulations on your appointment at the Allen Institute. We'll continue to see great things coming from your lab.
Yeah, thank you very much.
Right. Bye.
yeah. Thank you, Stephen. Thank you, Kate.